Hello, everyone. This is the African Society for Computational Biology and Bioinformatics, the monthly webinar of the System Administration community. We are here in a conference of Professor Go from Malaysia, who's going to talk about the recent advances of computational plant genomics. Professor, the floor is yours.
Okay, thank you, Abdullah, for the introduction. So, yes, everyone can see this is basically my background. I was actually not trained in genomics. I was trained in plant molecular biology. But then later on, because of interest, I self-trained to the next generation sequencing, and then established a group of plant functional genomics research group in 2012. So it's almost 10 years now. So basically, I'm going to stop my face recording so that it's smoother, just in case. So you're just going to listen to me. Okay. So today I'm going to share with you about recent advancement in computational plant genomics. Why I grade out the computational plant genomics, because the computational already part and parcel of genomics nowadays. So I could just say it's genomics. Okay. So basically, I'm from the National University of Malaysia, so it's among the top 150 in the world. And today the topic going to cover the genomics, and then going to talk about the current trend and future prospect with African context also, and also some part about precision biotechnology. If got time, going to just touch a bit about research and symbiosis. Okay, so for genomics, everyone knows genomics is let's just test your background, the history on the milestone of genomics. You can guess what are the milestones here. So first is the start of Mendel genetics in the 1900s. And then we have the discovery of the DNA structure by Watson and Crick with the X-ray crystallography by Rosalind Franklin. Then we have the Sanger sequencing invented in the 1970s, which allowed the Human Genome Project that lasted almost 13 years. So it's completed in 2003, but it's published in 2001. Then followed by this synthetic genome, okay, in 2010. So what come after 2013, where the CRISPR-Cas of genome editing. So it's already almost 10 years now. So what is it? Can you guess what is the latest breakthrough or the milestone in genomics? If you don't have answer now, but we'll come back to that again later, okay? So basically, in genomics is the study of whole genomes of organisms, whereas genetics deal with single genes or heredity. So it's commonly we familiar with the functional genomics and structural genomics. Computational genomics has evolved so broad until it become a field on its own. So what is computational genomics? It's the use of computational and statistical analysis to decipher biology from genome sequences and related data, which is a subset of bioinformatics and computational biology, the society today, okay? And it focuses on whole genome to understand the principle of how the genome control biology at the molecular level. So how it all started? It's all started in the 1965, around that, with this paper by Linus Pauling, a famous Nobel laureate in chemistry, that inference about the similar structure, similar function, and common ancestry based on sequence similarity. So basically, this is the foundation of all computational genomics. That the goal is to find pairs of similar sequences, just like the famous BLAST on NCBI. So the high sequence similarity imply evolutionary relationship, hence homology. Okay, whereas functional genomics deal with the expression of gene usage. And the most common one will be transcriptomics and proteomics on the transcript and the protein, but also it can cover until metabolomics. So for structural genomics, there are two definitions. One is about the physical nature of the whole genome. So for example, like this reference genome, mostly a haploid. And this, the example here given is the pan genome from deep sequencing of 910 African. And it deals with the mapping, sequencing, and annotation of the genome. When there's also the physical map, so like this karyotype, and then you can have this physical location of the genes. Deal with the genome structure. Now there's a 3D genome study. and also the comparative side of structural genomics that deal with the synteny, conservation, and variation. We'll come back to this again later. So you can see here, this is like how the chromosome synteny in the human genome. Okay. Whereas the second definition of structural genomics deal with the genome-based approaches with high throughput method like X-ray, NMR, or cryo-EM to deal with the structural determination of all the protein in the genome. So as an overview of genomics, you can see from the global comparative genomics from the fundamental side and up until the very applied translational genomics that deal with genome-wide association study or genomic medicine. You can see there's a broad range of it with a continuum and diverse field. So today, we're going to look at some of the trends in plant genomics. So these are the top 10 lists in my subjective opinion. So you might disagree with me, but I just want to introduce you all to these 10. So I'll go through one by one. So the first, the metagenomic, I think you have a session last 2 weeks ago on metagenomic for the oceans near South Africa, to the Arctic. And basic metagenomics is the calculation independent analysis of the collective genome of microbe within a given environment using sequence and function-based approaches. When it's deal with plant, then you can think about the soil microbiome or the microbiome around the rhizosphere or around the leaf, around the plant. So that's how it relates to the agriculture. And the road to metagenomics has come a long way since the establishment of microbiology with the invention of microscope. And it's all shoot up because of the NGS that allow culture-independent identification of microbe. And suddenly we realize there's so many microbes out there. And metagenomics is nothing new anymore, but now it's all moving from fundamental to application. For example, comparative metagenomics to inform the phytosphere for organic management, for the farm management. Also serve as an approach to detect and survey surveillance for the emergence of recurrent plant pathogen. And one example from the African scientists is on provide insight on the community structure and functional gene for the sunflower rhizosphere microbiome. Okay, so everyone is doing metagenome. So, but then the trend is going to application. And the second one, the evolutionary genomics, basically investigate the evolution of species genome to have the reconstruction of the ancestry. So as synteny I mentioned just now, it's basically the evolutionary conserved relationship between genomic region, whereas microsynteny deal with the relative gene order. In this study, using network-based microsynteny analysis, comparing 87 animals and 107 plant species, we found that the microclonality is much more conserved in mammals, whereas the with more single copy and synthetic among them, whereas the genes in plant are highly multi-copy and because of gene duplication. And we also familiar with this phylogenomic of the SARS-CoV-2 virus, and it's still ongoing, I think. You can go and get the real-time dynamic of how the strain evolved on this website. And you can see the proportions, how the genomic structure of the virus changed in real time since the beginning of the pandemic. Okay, third is the population genomics. We deal with the survey patterns in the genome within and among population to make inferences about evolution and genome. So we deal a lot with Bayesian statistics on population differentiation. A good example would be this malaria vector of the mosquito. To understand the evolution process affecting its genome, what is being selected, what the loci, and this population genomics trends towards the global level population genomic. With population genomic also come population resequencing. Okay, so the example here is the bitter gourd. So people study different traits of bitter gourd populations and do sequencing, get the assembly result and check the genetic structure to find out what is the history of the domestication of this fruit and what are the architecture underlying the domestication trait, like for example on the color of it, the length of it, or the texture of it. And with resequencing of many plant population, you get this pangenomic. Pangenomic is like combining all the cultivar, the variations, together like this as pangenome that include all the unique or the accessory dispensable genome. Pangenomes is in contrast to core genome, which is deal with all the essential genes present in all the different cultivar. So this pangenome can capture global and populational variation to link phenotype or interest to causal variants. For example, this recent publication on 26 inbreds of maize served as a founder for the nested association mapping population. So this allowed the identification of core set of genes as well as provide a pangenome for future study. And similarly, for this pangenome of rice accession, using the African rice to root the tree. So there's many can be done. And a lot of these pangenome deal with the structural variations and also gene copy number variation. Like this, using the Oxford nanopore technology long read sequencing of 100 accession of tomato, found this tomato plant structural variant genome that found many variations are related actually to the structural variation rather than the gene itself. So they use a genome editing tools to study the cis-regulatory structural variants and to identify the quantitative variation. And they found that, for example, when you CRISPR edit the promoter region, you get this quantitative variation rather than qualitative. For example, qualitative means present, absent, have or have not. Whereas quantitative is like the size variation. For example, there's a different kind of deletions of the promoter region resulting in different number, locule number, and hence different size of the fruit. So this is very interesting work by Professor Zachary Lippman in the US Coastal Harbor Lab. Okay, then with all the pangenomic combined from a different species, related relative, you get this super pangenomic. So it's getting more and more complicated in a way to combine more and more genome together. So you get this super pangenome at the latest trend to help with the functional study. And also number 5 is a genome-wide association study. To compare the genotype of large number of genes for large number of case, which phenotype, to look for the presence of variants of the gene, known as polymorphism. GWAS was first done on human, the age-related macular degeneration in 2005, so it's almost 17 years. And it has revolutionized genomic medicine by identifying the genetic risk factor for chronic disease like diabetes, heart disease, and cancer. And currently it's very well used among the crops to identify what loci, what genes are related for certain traits, and that can be used for marker-assisted breeding. So it's deal with the variant trait association. And this is how the overview of GWAS in plant. It can also deal with the metabolomics and also transcriptome to do the GWAS study. So in combination of everything as an integrated study. And the trend is moving toward meta-GWAS. So one GWAS panel is no longer sufficient. In this case, they use even 3 GWAS panel of 775 tomato accession with over 2 million SNPs from 3 panel to identify 305 significant association between the certain important traits that deal with the tomato fruit with the loci. Okay, and number 6 is the single cell genomics. And these are some of the technology used to deal with different type of investigation on a single cell. So this is for the plant single cell genomics. You can have a look on this paper, which is quite a good review on single cell plant genomics. And a good example of single cell would be the transcriptomic atlas. And this is an article from the recent issue on plant physiology. You can see this issue is all on single cell. So basically what they've done is they use flow cytometry with gene markers of different cells, and then they sort the cells using facts, and then they do the transcriptome sequencing for all the cell population from these different tissues, from the shoot and as well as from the root, to identify the gene activity in a developmental and embryonal context. So you can see the genomic field, the transcriptome even is going to like single cell resolution. Probably in the future or even subcellular resolutions of gene expression. So the trend is towards increasing number of using different technique and also dynamic integrated study using multiomics. So you can study the different lineage of the cells, either the state of the cell or how the cell in the trajectory of different tissues. Like just now, this different trajectory. Okay. So number 7, combining the single cell, you have this in situ spatial genomics. This means that you can actually study the gene expressions on the tissue itself, on which tissue, where the tissue express. So for example, this study on the in situ sequencing approach of the Plastocon 1, we together with 90 genes in the panel, they do the sequencing on the tissue sections. So there's at the same time, this is a Plastocon gene. They can identify how the pattern expression together with other genes associated with it, like GRAPIN. All these are important developmental genes. So they can actually look at the expressions from the sections and on where they are expressed using this in situ spatial sequencing technology. Okay, so number 8, the structural genomic that I mentioned, you can actually study the genomes at different levels. So up until the DNA resolution to the compartmentation of the chromosome, you can use different techniques. So if you are interested, you can go and look up to this review for explanation of the high-C sequencing and etc. So it deal with mapping the 3D genome architecture and study whether this genome structure affect the phenotypes or not. And I think in the human cancer, they do detect the structure, the genomic structure actually affect whether the cell going to be cancerous or not. But in plant, this kind of study is still limited. So there's a company called Spatial Genomics deal with this. So it's quite interesting that they do this sequential fluorescence in situ hybridization sequencing. Okay. So number 9 will be the system integrations based on network analysis. So it provides a platform for inference of different omics data. So many network methods have been developed mostly for the cancer, but also for the for plant as well. But still, if compared to cancer study, I think it's still lacking for the plant counterpart. So for the plant multiomics, also known as panomics, they deal with the integration of multi-omics data, for example, to study leaf senescence in this case, or pathogens effect. And this is an example of how you use to identify or discover plant signaling molecules using the multi-omic approach for integrated with between metabolomic and also the whole genome co-expression analysis to identify signaling metabolite, either known or unknown structure. So combining single cell genomic and multi-omic, you get single cell multi-omics. So yeah, the field is getting into more integrative. And why do multi-omics or panomics? Because the molecular complexity at different level is different. The closer you are towards the phenotype, require you to look at the interactions like this interactome. So just simply looking at the RNA transcript alone won't be sufficient. That you have to look at the proteome as well as the interactome. So multi-omic allow measurement of different factors and the phenotype together to better connect this variant with insight beyond a single omics. So it also provides a holistic view of the biological system for increased discovery power. So it deals with biological discovery. And there are various approaches for multi-omics integration, either at the elemental level, pathway, or mathematical base. So you can have the correlation between gene expression and protein expression up until genome scale analysis. And from a field application point of view, there's either genome first, phenotype first, or environment first, depend on the purpose of the study. If you are gene-centric, then it's known as genome first to identify the GWAS loci. Or your phenotype first, you want to look at what trait, the focusing, what is causing that trait. So it may be multiple to understand via the pathway contributing trait, or the environment first to look at what's environmental factor actually interact with the genetic variation. So this kind of approach is needed to apply finding to the field for value creations. So such have challenges also because you need high quality sample material for omics study, and you have to have technical expertise for different omics, and you need the technological demand for high resolution and sensitivity. So with that, the instrumentation need funding and it's expensive to do this research and development. Also for commercialization and innovation. And there are also an uncertainty for field application. So lastly, this is the most exciting of all. And the answer to the first question that when I asked you about the latest breakthrough after the genome editing. So I think some of you already know the answer now. Okay, so it's the application of AI. So AI in genomics is I think the latest, most exciting field, and using deep learning and combined with computer visions allow you to analyze a lot of genomic data by solving classification, segmentation, image retrieval problem. Also, in combination of natural language processing, it allows you the researcher to analyze all the publication related to genomics for information retrieval. Okay, some of the AI applications include identification of genes, predict protein structure, function, association, as well as binding, and there are many things. And the latest breakthrough will be this DeepMind, the AI breakthrough, which allow the whole structural genomic to be created based on sequence. You can have the structure function study. In the past, you have to clone the enzyme, clone the protein, and then to do X-ray crystallography or cryo-EM to study the structure, to infer the function. But now, based on the sequence alone, you can get very high score of accuracy of the protein structure that you can straight away, like near to experimental quality. So you can actually infer function based on the sequence. So this will be the latest breakthrough. And also the AI in plant genomics, deep learning along the central dogma by identifying the genes towards the phenotype, doing correlation, regression analysis, as a flow of information in making sense of the genomic variation from association to causality and molecular mechanism. And this is another example in Arabidopsis and also maize to study the transcription factor connected to the nitrogen use efficiency by using a machine learning approach. If you are interested in this topic, you can look at this review, and I think it covers most what I want to present. And from genotype linking to the macroscopic, so it's also require you to have this other technology beyond genomics for the field application, especially for phenomics, the phenotyping of all the traits. So in relation to that, you can combine the GWAS study with the hyperspectral reflectance information to correlate their relationship for machine learning prediction. So now we are moving towards Industrial Revolution 5. What it means? It goes toward personalization. Everything is precision personalization. The industrial IR4, the fourth one, is all about automation, the digitalization. Now it's about smart, precise using AI. Okay, to summarize this part of the talk about the trend, genomic research has largely driven by sequencing technology. More data were generated by third generation sequencing like nanopore and also PacBio. Then we have large-scale global population genomics, pangenomic and single-cell genomics become more common with research consortia. Then GWAS will continue for the identification of polymorphism, and is moving toward meta-analysis. Multiomics system integration of biological big data and network analysis provide a more holistic framework for convergence of genomics. So genomics also going towards convergence with other technologies. An automated analysis pipeline using AI-driven technology analytical tool will become useful to accelerate genomic research. And there's an increasing emphasis on applied and translational genomic research. Okay, And just a recap, our top 10 trend, all the metagenomic until pangenomic are resulting in integrative study together with the GWAS and One Health. And then the single cell in situ structural and system integration get this spatial panomics with automation and personalization with AI together, convergence into translational to achieve the UN SDGs. Okay, so all have to be applied for well-being of the planet, people, and prosperity. So for the latest breakthrough, you can go through all these journals. And now I'm moving into the second part of the talk, which is the prospects. What are the future prospects? You can see that plant genomic market is on the rise with 8.3% that worth 11.7 billion by 2025. And this increasing demand due to the integrated crop management using plant genomics to ensure better crop management. So the demand in the market is to inform crop management. But then this is compounded by the high complexity in plant genomes. So it's not easy doing plant genome research. It's very costly nowadays. Okay, and then also another attractive opportunity related to genomics is the plant breeding and CRISPR market, which is higher in the growth, 14%, that will be worth 14.6 billion by 2023. And there are other applications of plant genomics that you can refer for breeding, for genome editing. So all the genome help with genotype, contribute to genomics like the genotyping. This contribute to a better crop with low input of agrichemical input, so that we can have a better crop to feed the world. Okay, so artificial intelligence or bioinformation out there, there's a future for you if you are familiar with this. And the predicting disease with AI validation can apply the same framework to crop trait yield or disease forecast. So if you are a biologist, on the other hand, you should become computational because this is a minimal skill set for you all to be familiar with the Unix Linux system or the scripting language like Python, and then database creation using MySQL or practicing some of the computational technique, especially on statistical programming using R. So some of the opinions from the biotech leader, they also think about super resolution imaging of the genome, single cell, and then precise large-scale perturbation, this is like genome editing, and then omics coupling with synthetic biology, single cell multiomics dimension, and genomic surveillance. Okay, so more on synthetic biology and also DNA encoding. So this is a good statement about this, that a better understanding of genome structure and molecular basis of trait will help to develop improved crop variety to address climate change. So, but then I think everyone's aware that the genomic data is increasing exponentially. So one day we're going to run out of the space, physical space to store all this data. So probably in less than 200 years, this will happen. So what the future storage? So it's just while thinking that there could be DNA computing in the future, in the long future, because DNA is so high in the density of storage, that the data storage is almost twice of hard disk. And this is another possibility that data will be stored in the silicon for archiving. You can see just the singles, you can store the whole Holy Bible in just a chip. So it's to space saving, but to read them could be difficult. For example, DNA can last for billions of years. So it can be a very good way to store or archive certain data in CGTA. So let's come to Africa context. So we see in general, the genes, the sequencing all go towards the third generation. The first generation are all lower quality with lower contiguity of N50 and also smaller genome size. And from this review of the past 20 years, there are almost 800 plant species now with genomes. And the trend is towards all these like long read sequencing technology And you can see some of the model plant have so many assemblies. So this from the population resequencing I mentioned. And among the 798, only one was submitted from Africa, from Nigeria. The others mostly from China and USA. And a lot of crop plant local to Africa actually was sequenced by researchers elsewhere, as you can see here. And in Africa, actually, there's a large population with large agricultural related land use of over around 70%. You can see it account for 1/4 of the agricultural land in the world. And for the cropland is like 1/5 of the world. That means most of agricultural land is actually used for animal farming, for the pasture and meadow. And comparing between Africa and Asia, we can see the big difference on the focus of the planting plants. Like for example, maize is the top area for planting, whereas rice in Asia is the most. Whereas total production, cassava is ranked the top in Africa compared to rice in Asia. This relates to the orphan crop, which is the potential. I think when talking about genomics, people are thinking about orphan crops, Africa with little attention. And this orphan crop is actually very important to provide gene pool for future crop improvement. So, and also for future proof the crop for diseases. And this is a framework on how it can be used to combine from major crop and new crop with orphan crop information to improve future agriculture by providing genetic resources to identify functional genes, also study evolution on and use the genome-based breeding technology to do so. And one example is this pearl millet genome sequence for the arid environment growth. And there are, I think, 100 orphan crops listed in for the African orphan crop consortium. I'm surprised to find papaya also one of them, and also mangosteen, which I work on, also one of the orphan crop. Okay, these are some of them, and there are actually many big organizations was involved, including the BGI, and some of these actually are like, for example, ICADA doing system research for comparing different system of farming, and this potato center also relevant. And then this International Crop Research Institute for Semi-Arid Tropics doing different legume, and this maize and wheat improvement center. So the latest publication from the plant orphan crop are these 5 species, but they are not from the African researcher. Only 1 or 2 are affiliated, but they are not actually African themselves, I'm not sure. And this is the latest review by Abdullah, so your host today. And I think there are around 13 was like sequence African plant genome. I think most of them are not, they are just African-based group that sequence them. And most of them are based on the single short read sequencing. I think only one using the PacBio for the to enhance. So especially for the transcriptome also using short read. So when I search for the document related to plant genomic and plant genome, there are around 21,000 documents. Among them, 439 belong to African country, and the most is from South Africa, leading. Then is Kenya, and the author with the most is this one Dipier. So with different formatting of naming. And University of Pretoria, I think that the previous speaker also from the same university are also good in also reputable in genomic field. So quite some high profile population, but mostly from affiliated researcher instead. But there are some rising local plant genomic project like this Eucalyptus grandis by the local African scientists themselves. So there are many opportunities, and some of them are like DNA barcoding for biodiversity of plants in Africa, some of the herb genomic for animal medicine, novel drug discovery of nutraceuticals, and also metabolic pathway bioengineering, and also precision biotechnology. So I think there's a rising hope for African scientists, and this is a recent editorial, I think, on the female scientists. And to bring everything together in perspective, We know the genomic data is increasing exponentially. Storage will be a problem. So, and everything is going towards cloud computing at the moment. The bottleneck is on the functional validation, data curation and analysis for meaningful application. So the wet lab automation and AI and machine learning will be the current effort. More and more people or crop will be obtaining on whole genome sequence. This allows precision medicine for the human, and precision agriculture will become a reality. When genome combined with epigenome and microbiome, you result in integrative healthcare or soil health. And there's an expanding global market for the field of genomics. And with this, we need a new generation of biologists with computational expertise and experimental skills. And there are important roles for bioinformatics in curation, management, and analytics. And for those that cannot afford to do sequencing themselves, I think there's a lot of opportunity for data reanalysis. Actually, the group can use new tool or software approach to study or to use plant genome reference to reanalyze previous study or do meta-analysis for further insight or new insight from the previous studies. So you can reanalyze the data. Also, plant genome science has arrived at an exciting time. There's increasingly diverse group of scientists. Everyone should embrace long-read sequencing technology for genome sequencing, and there's a need for assembly for clades, the plant clades with null or complex genome. Collectively need to monitor the large-scale consortia collaboration project to ensure ethical approaches with in-country people giving a voice. And participation and access to resources should be broadened at every level to empower researchers across continents regardless of social-economic status. And then don't forget for public engagement to foster equity or equality in STEM. So agriculture has evolved with genomic advancement. So like I mentioned, the start of the genetic Mendel's lead to crossbreeding because of the resulting in green revolutions, together with mutation breeding, and then lead on to gene transfer through modern biotechnology, which is a gene revolution. Now we're moving into precision revolutions of precision genome editing. So with the genome information, this allows a lot of marker-assisted breeding to circumvent the biosafety issue. So genomic, how it contributes for precision biotechnology by going to GWAS, high throughput genotyping, phenotyping, and functional genomic, allowing molecular genomic assisted breeding or genome editing for targeted. So this leads to a precision agriculture. And this cannot be alone. It has to be integrated with the technology, other technology like ICT or Internet of Things, drone, imaging, machine learning, and robotic automation together in the fourth IR. So for crop improvement in precision biotechnology is not something isolated. It's all the aim for people to get these desired crop traits. And how to do so? People actually do population resequencing that I mentioned, and then do it through targeted way via genome editing or naturally via crossbreeding that take longer, much longer. And some of the recent genomics publication that allow this kind of precision browsing, including this pigeon pea, chickpeas, and in Malaysia we have this oil palm genomes, and this well studied on the different genes related to the trait. For example, the virulent genes related to fruit color, and the shell gene for the kernel oil, and then you have this karma, the genes related to epigenetic deformation of the culture. So these three already applied for diagnostic to save a lot of losses. And there are other traits actually ongoing. And the goal for the crop industry actually to make more profit. So that's the reality. For all the research you have to apply to the industry, and for them is to increase yield for better profit. And we want a more sustainable agriculture by having a better plant materials. Okay, and also better products. And this requires integration and digitalization with the various platforms on cloud computing together with the genomic technology. And this kind of transformative technology includes different fields from different areas. So it requires convergence of technology. And genome is just one of the technologies. So we have to think in this kind of context for precision agriculture, not just genomic alone. So I don't think I have much time. Just briefly go through what we do in Bioethics. Bioethics is the first and only institute of system biology in Malaysia. It's like almost 17 years since its establishment. So we use system biology to integrate all the different omics to review biology of plants and microbes in focus. We use system approach for biomolecule discovery of this plant, including this carnivorous pitcher plant that I work on. Then you have this agar wood for the premium resin, and you have Jatropha for the biofuel, and then we have kratom for the and also kasum, our local herb for its 60% of flavonoid, and it's famous in the local cuisine. Also for crop improvement of the fruit and rice. And we have other plants also like firefly, also the pest of the oil palm, and also tiger grouper of delicacy and expensive fish. So we use this framework of multiomics and tissue culture technology to towards synthetic biology or crop improvement for commercialization. And this is a framework that we use. And actually, if got time, I will have gone through these 3 examples of you. But however, for those that are interested, you can join the webinar in May. I'm going to talk about these 3 examples in the future. So for now, I think I'll just straight away jump. Sorry, I turn off the laser pointer. So I'll just jump to the conclusion, the take-home message, because we are running out of time. Just 1 minute left. So precision biotechnology will accelerate crop improvement. Established local in vitro technology can be leveraged for advancement of genome editing. Genome edited crops still need to overcome the hurdle of biosafety regulation and public acceptance. So translational research while genome assisted breeding will be more promising in producing improved variety. And panomics integration is becoming a common approach. Convergence of biotechnology or genomic with ICT and IoT will realize the 4th industrial revolution. And with that, I thank everyone for attention. Just to introduce to everyone, we have I'm going to organize a workshop. This is online, so everyone from everywhere can join if the time zone allowed. So it's in July. So anything, you can follow our group Twitter, also join the Facebook group, called Plant Functional Genomic Group, and also visit our my group website for more information. I think with that, I thank everyone for attention, and I welcome any questions. Okay, Abdullah, back to you.
Thank you, Professor. It was great. It was really great, actually. You have all the trends on plant genomics in the world, and especially in Africa and Asia. That was really great. Thank you very much. We have two questions here. I'm gonna just start by one. Was by Fusborn. Then we're gonna go to Ali. He say, why the implementation of genetically modified organisms still in challenge in Africa? Even the genomics works in Africa, and it really leads to a sustainable agriculture.
Can you repeat the question? It's like
the question is about the implementation of genetically modified organisms. You say they are still in challenge in Africa. They are still not working in Africa. Even genomics is working in Africa. So why do you think?
Why do I think it's lacking behind for Africa? Is it? I think it's the acceptance for the genome modification. Even in Malaysia, we don't have GM crop.
Yeah,
it's public acceptance, and it's a problem with it's not the technology itself, but people prefer public prefer natural fruit, organic crop, organic food. So that is the public acceptance, and in terms of that's why in terms of the funding as well, it affects. When people think about there's no future if you work on this kind of GMO in your research in Africa, why would someone fund it? Why the funding agent should support you if there's no product at the end? So and so if you cannot bring the product to market in the first place, why should you do the research?
Problem more than a research problem.
Yes, it's more a societal problem. At the funding agency and among the researchers themselves. I think in Malaysia we also have the same issues. It's not just Africa.
Okay, thank you. We have another question. You can open the chat. There is a question there. One by Awi.
Okay. In the future of omics, can AI or machine learning stand alone without lab experiment to confirm the prediction, i.e. after the machine learning model are trained on omics data set and they are able to make prediction? This prediction is still need to be validated experimentally in the lab. Could wet lab really be automated? In terms of wet lab automation, you can actually, a lot of microbial culture is automated. You can screen many genes function in microbe using automation. Like for example, you can do automated PCR, automated screening, chemical screening, automated bioactivity assay. So all those wet lab can be automated for large scale screening. And also the first question that whether the AI and machine learning can stand alone. Okay, without validation, I think those are all just hypotheses. So unless the AI really established to very high precision with very low false positive or high precision, then I think you still need to do the validations. Because if even the training data set itself is based on the population. The African population is different from Asian population. So the training set depends on what the population is used. So there's a limitation still, and there's I think a lot of work ongoing for the regional effort on the different population for the AI prediction. But as of now, you can see that in terms of the AI for protein structure prediction, that is very high precision. I think people can straight away do that to generate hypotheses about structure function study. So it also depends on the application, whether it should be standalone or not. If let's say it involves life and death, probably you shouldn't just rely on AI prediction alone, right? You need to do something to monitor, or before you just all decision depend on the AI outcome. So there's this like movie on the future of AI that become droid that decide whether someone to live or not. So that depend on your genome. That will be crazy, you know. This there will be a big issue on the bioethics, bioethic 3.0 or bioethic 2.0. Like when you were birth, you already know your genome. You know already your risk factor. What is your risk of getting this disease? Should you be alive at all, knowing that you're going to die in 20 years' time?
I think from the ethical problem, there is another thing is that genomes doesn't contain everything. We cannot know all information from genomes. Like for now, you just talked about multiomics. So
Yes.
we are now talking about different layers of the of the omics, not just genomes. We are talking about proteome and what it can, how it can enhance the phenotype or I don't know, deactivate some phenotypes. We talked about epigenomics, microbiomes, and microbiome is still an ongoing subject. So far, there is a lot of research in the microbiome, and and we are still discovering this new layer of the genome, which is a genome of bacteria or archaea that lives inside us, and until the 21st century, we still believe that they might be pathogenic, but now we know that they are part of our organism. So I don't think that relying on genome alone can really give this kind of information about living or death.
So
Yes.
what's your question? That's a comment, is it?
Yes, that was yeah, that was the comment. Actually,
Yeah,
I
just
have
follow up on your comment. Actually, I think the more we study, the deeper it goes, the more complex we realize about biological system. That the more work needs to be done. Never ending of discovery.
and we just unveil some new layers of what we can study, and we got on trouble more and more.
we get into the deeper and deeper because we want to predict, but then it end up that the prediction is wrong, so there's something not right. Then we have to go deeper and deeper.
So I think
But actually, the model itself provide a good framework for people to investigate further.
actually I think yeah, I think if we talk about precision and production, I don't think that in terms of eukaryotes we are there yet. I think we are way far from there. Yes, we can do some kind of personalized or precision in some things, but not in the whole sector. Like for example, uh, you're talking about bacterial, and I think for bacteria the thing is that we have a lot of data. So yes, we can go to precision there because we have a lot of data, so we have a lot of information. But talking about plants or animals or fungal, I don't think that we are there yet. I think we're still far from that point of precision or personalization or even
Yes,
protection. We still need a lot of
but
data.
the goal is towards that.
Yeah,
That is the
exactly.
goal. Just
Still a
like
goal.
now we have the 4G technology, the 5G, but now people are working on the 6G.
Yeah, yeah, yeah.
China
So,
is working on the 6G. So when we are still catching up with the fourth IR, people are moving towards the fifth IR. So we have to always aim forward what is happening next. So what's happening in 2030? What will be achieved? What will be the next breakthrough?
I think we are going in some kind of exponential way. So probably what we have reached in 10 years gonna be reached in 3 years probably or 2 years in the future. Because we are going in an exponential way, not in a linear way. So I think things gonna be going very fast in the future compared to the last decade.
Yeah. So can AI save the world?
But yeah.
Actually,
um,
I written a commentary on the AI to regulating AI for the SDGs. If interested, can have a look. There are actually many problems with AI. When you are technology itself is fabulous, but when you use in a human context, there will be a lot of misuse.
yes, of course, yeah, yeah.
Yeah.
That's true. That's true. That's true.
But in terms of the research wise, our constraint is always the human researcher, also the funding. So how you get more funding or money to run the research. As the research is getting more and more high resolution, you need more money to do all this expensive work.
It's kind of a paradox.
Yeah, so probably in the future you might everything become automated, that you can just put in your samples, extraction all done automatically, sequencing, sample, library preparation all done automated. Then getting the sequence, then the assembly, everything is automated. It's all like a pipeline. So one day you can actually have many genome sequence.
At a minute.
Yeah. Or you can real time sequencing. Like now the nanopore sequencing is going towards the real time. So in the future, you can just give a drop of blood and then go to a clinic that give you these precision prescriptions just for you, personalized genome.
It will be. Actually, I think I will ask something about the webinar that are gonna be in May.
Oh, the link of the webinar, there's no link yet, but you can go and follow the Bioethic website. So there's a Facebook on my institute, the Institute of System Biology in Bioethics, and we're going to advertise on there. And I'm going to share with Abdella nearer to time when we have the date
Perfect. Thank you.
established. But I'm going to share on the Facebook group also, on the Plant Functional Genomics group nearer to date. So you can actually join the group and have a follow-up.
Thank you. Actually, I have a
Okay.
last or I will say last two questions probably. One is about the genomic trend from an economic perspective. Like so far, we are talking just as researchers, but people are not just interested in research; they are interested in money too. So, do you think that in Africa or in Asia, we can talk about the rise for the future startups or new startups in genomic field in any procedure, in any process, not just in like the Illumina or Pacteo, the sequencing platforms, etc. Can people really start making startup in this field, in genomics in general, or in plant genomics especially?
It will be very difficult if you want to have a startup solely based on plant genomes, because most of the plant genomes, as we can see, have commercial values, and it's actually done by championing institute. For example, Erie for the rice, and then in Malaysia we have the Malaysian Palm Oil Board that do all this. They spend a lot of money to just sequence one commodity crop in Malaysia, and you have this rubber tree also of commercial commodity. But in terms of orphan crop, why is important to have this consortium? Because you manage to get a group of people to raise funds and to justify the importance of it. But instead of making it into a commercial, will be very difficult, because how are you going to patent the genome? You can only patent the applications that you learn from the genome. Like for example, for the oil palm, it's a diagnostic kit. You cannot patent the oil palm genome, but you can patent something to generate income and have a startup that derive from the genome itself. But in terms of having a startup from the genome itself is difficult. You can have the startup from the application of the genome information, like diagnostic kit, marker-assisted breeding, or diagnostic agronomy company. Things like that probably is doable. But as a startup for just purely genome sequencing will be impossible, because it's too expensive, and the ROI, the return of interest will be, investment will be too difficult to forecast.
Okay, thank you. Any other question? I have one last question. It's actually kind of I don't know. Do you think in terms of multi-cell organisms like plants and animals, do you think sequencing one tissue or is enough to decipher the whole genome of the organism? Like, don't you think that it might be some variation inside the same organism, some differences in the genome inside of the cells that belong to the same organism, to the same individual?
It based on the context of cancer cells, this matter.
Okay.
Yeah, but in plant, the cell are static. So the genome mutation between the cell, between tissues, if it's genome, be very little variations. But in terms of the mobile human cell, probably that matter more in your context, like different tissues, whether the genome. When we're talking about genome, most of the time we assume that is the same, but the epigenome changes. That's why people are more keen on epigenome if it's the plant, because it involves the regulation of the expression. So it's going towards the functional side that lead on to the phenotype. Whereas the genome itself just a blueprint. We want to understand the mechanism, then we have to look at the functional aspect, including the epigenome and also the functional genome, like proteome, metabolome. Whereas in cancer cell, the genome changes. So even the fusion gene, the genome structural variation, structural changes, chromosomal rearrangement, all those matter even between cells. So that's why single cell study is so important for humans.
Thank you very much.